Good day, my dear colleagues in pediatrics. It is my honor and pleasure to be in front of you today to talk about a very important topic. And these are our learning objectives for today. Number one, I hope to give an overview of brain development and neurodevelopmental processes in the early years. Number two, we will discuss importance of myelination in cognitive development. Number three, we hope to be able to cite modifiable factors that affect brain development in general, with focus on relevant studies on myelination and nutrition. And lastly, we hope to discuss salient points of a most recent study on the effect of nutrition on developmental myelination on term infants. Allow me to start with this illustration of a scaffold. If we are to build a house or building, we know that scaffoldings are built before the permanent parts of a structure are put in place. Scaffoldings should be very sturdy in the hope that this comes down when the structure grows stable and more secure. Parenting is akin to that, in that we provide our children the necessary elements for growth and development as we mold them toward resilience, productivity, and independence. In his book entitled Scaffold Parenting, child psychiatrist Dr. Harold Koplowitz of the Child Mind Institute talked about how this deliberate stacking of building blocks for an effective scaffold from infancy, which are then gradually loosened one at a time as a child gets older, is possibly the single most effective way to encourage kids to climb higher, to try new things, grow from mistakes, and develop character and strength. Modifiable factors like stimulation, proper sleep, and nutrition further strengthens the foundation to help develop a healthy body and a resilient and integrated brain. Early childhood is a very critical period of growth, which includes physical growth, social and emotional growth, and of course, cognitive growth and learning. And this is the time when we build those scaffolds. There is no doubt that along with environmental influences and social interaction, nutrition plays a key role in brain development and learning. This graphic allows us to more specifically examine the interplay between these factors and how they eventually influence brain and behavioral development and cognitive outcomes. Saying that, the first 1,000 days in a child's life is a critical period not only for the physical or anatomical milestones that are taking place, but more importantly because of the dynamic changes happening in the brain as early as conception. In this adapted visual of the timeline of key human neurodevelopmental processes from an article by Silberys et al., we see that the earliest developmental process of neurogenesis, or how the name implies the development of nerve cells or neurons, as early as the 8th week post-conception. Cell body, dendrites, axons develop during this period and peaks during mid-gestation. The neurons then group and migrate to different areas where they make rudimentary neural networks. This next slide gives us an overview of the three other vital neurodevelopmental stages, apart from neurogenesis, which we already talked about. These processes are shown to contribute to cognitive function, including learning and memory. Synaptogenesis is the formation of synapses between neurons in the nervous system. The first synapses are produced very early on by week 8, but synaptogenesis is not overly active until week 24. This process accelerates drastically after birth and peaks by the second year of life. When that happens, many neuronal connections become exuberant. They provide room for the most important networks of connections to expand. The connections, however, that are not used often are eliminated through synaptic pruning, which can continue into the early 20s. Connections can form throughout life, and the unused connections, continue to be pruned. Lastly, Myelination provides the foundation for brain connectivity. It is an expression of the functional maturity of the brain and has been shown to be associated with cognitive performance, memory, learning, and skill development. This process starts as early as gestation week 12 and continues even into adulthood. Myelinated neuronal regions of the brain or white matter volume increases in accordance with age. Myelination is a cornerstone of human neurodevelopment. It ensures coordinated communication between brain cells and networks. It is positively correlated with cognitive development throughout postnatal development. In a study performed by Schidtmorst and colleagues, general cognitive function correlated with white matter architecture. Another study demonstrated improvement in the cognitive test scores for receptive language that corresponded to white matter maturation in breastfed babies. It has been shown that symmetrical white matter distribution correlates with language, reading, and memory. Some research also demonstrated a parallel between characteristics of white matter tracts in infants and their visual working memory scores. Speed of cognitive processing abilities was shown to be closely related to the structural integrity of white matter pathways. In a recent interesting preclinical study on mice on long-term memory and myelination, scientists from the University of California in San Francisco have discovered new myelination forming within the brain during long-term learning, specifically in motor learning, like when the mice learn to run on complex wheels, for example, and in spatial learning when mice, for example, try to find their way in a peculiar or specific location within a maze. They hypothesize that new myelin formation may play a specific role in the consolidation and maintenance of long-term memory. For developmental myelination to take place accordingly, the assembly and maintenance of the myelin sheath requires a carefully orchestrated delivery of nutrients that include lipids like polyunsaturated fatty acids, phospholipids and sphingomyelin, proteins, and a host of vitamins and minerals. During the first year of life, approximately 60% of an infant's total energy intake is used by the brain to synthesize neuronal membrane and myelin sheath. Prado and Dewey, in an article published in Nutrition Reviews, cited that several factors influence whether nutrient deficiencies during this period of rapid brain development cause permanent cognitive deficits in human populations, including the child's interaction with the environment, the timing and the degree of nutrient deficiency, and the possibility of recovery. Nutritional deficiencies from nutrients like iron, B12, folate, iodine have been associated with hypomyelination, altered myelin composition, and decreased myelin synthesis. Early life differences seen in fatty acid-related white matter composition, myelination, and cognitive abilities among breastfed and formula-fed infants are linked to long-chain polyunsaturated fatty acids, sphingomyelin, iron, and folic acid levels. Myelination, as we said, is a cornerstone of human neurodevelopment. Again, it ensures coordinated communication between brain cells and networks. And acknowledging the importance of developmental myelination and its impact on learning and behavior, along with advances in neuroimaging, scientific professionals have set their sights on the research goals of the implications of nutrition on developmental myelination. There are some pioneering studies in this field. To start with, there is this study of Tanaka et al. On sphingomyelin, which is a pilot randomized controlled trial on low birth weight infants that showed increased sphingomyelin plasma and erythrocyte levels at 4, 6, and 8 weeks of life, along with improved scores in behavior and attention measures at 18 months following intervention with sphingomyelin-fortified milk formula. Myelination improvement as a possible mechanism was cited by the authors for this. In another study about DHA, preclinical rat model done by Hobner et al. Reported DHA effects on myelin composition and function in a concentration-dependent manner. In yet another study by Hauser et al., in their in vitro study, they showed that there was an increased number of oligodendrocyte precursor cells and also differentiation and maturation in primary cell culture growth given the blend of nutrients that we can see here in this slide. Significantly, the findings were only seen in higher doses. The interaction effect between investigated nutrients with positive net effect for the blend were not observed in the individual nutrients. However, to date, there is no interventional study that has investigated, at least de novo, the effects of nutritional supplementation on developmental myelination. Observational studies suggest differences in developmental myelination between infants fed differently. From that premise, this study, authored by Schneider, Bouchage et al., entitled "A Nutrition Formulation Affects Development Myelination in Term Infants," a randomized controlled trial published in February this year in the Frontiers in Nutrition journal, the researchers put forth the following objective. They set out to investigate the efficacy of a blend of DHA, AA, iron, B12, folic acid, and sphingomyelin from a uniquely processed whey protein concentrate enriched in alpha-lactalbumin and phospholipids versus a control formulation on myelination, cognitive, and behavioral development in the first 6 months of life. The hypothesis was higher levels of the nutrient blend increase myelin in the developing brain. The study is an ongoing prospective, longitudinal, two-center, double-blind, randomized controlled parallel group design across a 12-month intervention period with follow-up assessments up to 2 years of age. The subjects underwent 7 visits either at the Memorial Hospital of Rhode Island or the Pennington Biomedical Research Center. 7 visits during the intervention and 2 visits during the follow-up period were done. Reported findings in this paper are for the 3 and 6 months follow-up. For 6 months staged analysis, a clinical site collaborator and external research organization were unblinded only to aggregated information so that this can be shared to ensure blinding until trial completion. The sponsor adhered to the WHO code supporting breastfeeding. The study team did not influence parental feeding choice at any time, and a lactation consultant was freely available to support and advise mothers regarding lactation. The subjects were enrolled within a window between 2 to 5 weeks for formula-fed infants, and the breastfed infants were enrolled earlier. The table on the right details the nutrient levels in the formulas used for the investigational and control group along with the reference standard. Trial intervention used bovine milk-based infant formulas that were taken orally for 12 months. For the control group, the formula contained standard whey protein concentrate, while for the experimental group, the formula contained alpha-lactalbumin enriched whey protein concentrate with higher phospholipid and sphingomyelin levels, and it also contained higher levels of DHA, ARA, iron in the form of ferrous sulfate, and folic acid, B12 in the form of cyanocobalamin versus the control. Reference group samples were collected at visits 0, 1, and 2, and the analysis was done anonymously. We have to note here that the higher levels of nutrients were based on observational data and adjusted based on regulatory requirements for infant nutrition products. Both products, the control and the investigational ones, had the same organoleptic and sensory characteristics to secure blinding. MRI was done during natural, non-sedated sleep for whole brain and white matter regions of interest such as the cerebellum, the frontal, the parietal lobes, the genome, the splenium, the occipital, and the temporal lobes. For this stage statistical analysis, the trial outcomes are as follows. Myelin measures from MRI at 3 and 6 months, myelin volume through myelin water fraction or MWF. MWF has been histologically validated as a specific marker for myelin using human tissue and animal models of myelin damage. The higher the MWF, the bigger the myelin volume. Myelin structure was also measured. Additional outcomes such as overall cognitive development using Bayley 3, socio-emotional development using the Ages and Stages questionnaire, the socio-emotional component of the second edition, brain volumetric outcomes, infant sleep using brief infant sleep questionnaire, and descriptive measures that included adverse events and safety assessments were also included. Children were randomized into one of two intervention groups, either receiving a blend of DHA, AA, iron, vitamin B12, folic acid, and sphingomyelin from a uniquely processed whey protein concentrate enriched in alpha-lactalbumin and phospholipids, or a control formulation. Non-randomized breastfed children served as a natural reference group. As illustrated in this flow chart, a total of 189 infants were enrolled, of which 81 or 42.9% were randomized into the two intervention groups, and 108 or 57.1% of those who participated were in the breastfeeding or the reference group. The mean infant age of enrollment was 29.9, and baseline characteristics were similar between intervention groups. The full analysis set comprised 174 children, and the reasons for premature withdrawal were evenly distributed between the randomized groups. Imaging data were available for 99 infants at 3 months and 71 infants at 6 months of age. These were, however, reduced to 91 infants at 3 months and 66 infants at 6 months of age. This was due to operational constraints because of COVID that stopped recruitment at 81 infants for both arms and 108 for the reference group. Parent-reported adverse events and infant growth measures were collected, along with medical history and serious adverse events. These composite images show the regions with higher myelin water fraction or MWF and thus myelin volume at 6 months. MWF was statistically larger in the whole brain, cerebellum, parietal, occipital, and temporal lobe. As for myelin growth rate, there were no significant intercept differences that were found between the three groups. For brain volume, behavior, and safety, there were no significant differences found for total white matter, gray matter, cerebellum, nor for cognitive language, motor, and socio-emotional development, and sleep at 3 and 6 months of age. Safety findings were similar across groups, with weight and length that were between 10th and 90th percentile for most infants. There were also no serious adverse events reported. Parent-reported constipation as adverse event was reported across the different groups, but were slightly higher in the investigational group and will be further monitored as this study proceeds. A very interesting finding is the reduction of night awakenings at 6 months by 38.6%, or in more than 1/3 of infants in the investigational group versus the control group. This was based on the number of night awakenings as assessed by the Brief Infant Sleep Questionnaire or the BISQ. Though statistically non-significant during this time point, it will be very interesting to see how this parameter will evolve until the study ends. Generally, newborns sleep a total of about 8 to 9 hours in the daytime and a total of about 8 hours at night. But because they have a small stomach, they must wake every 4 hours to eat. And most babies don't begin sleeping through the night until at least 3 months of age. So this is a very interesting finding of this study. In summary, this is the first study to demonstrate the impact of myelin nutrient blend on developmental myelination in term infants as early as 3 and 6 months of life. Increase in myelin volume, whole brain, parietal, and temporal lobes at 3 months, along with cerebellum and occipital lobe myelin volume increase seen in subjects given the myelin nutrient blend. These regions are known to be functionally involved in sensory, motor, cognitive, and language functions. Number 3, there were no measurable group differences in cognitive and behavioral outcomes that were observed this early, as similar with some previous reports. As some final vignettes to share, I would like to share these important ideas to my pediatric colleagues. Number 1, we know that nutrition is the substrate for normal physical growth and development in all tissues of the body, including and more especially the brain. Modifiable variables like nutrition and environmental factors are positively influenced by proactive and preventive behavior and practices. And so as we continue to strive to safeguard the future of our children in the way we live and work for the betterment of our country, our role as pediatricians, gatekeepers, and counselors in optimizing nutrition is vital in protecting overall health, especially brain development in the early years of life. Thank you.